welcome to the first video lecture for Advanced Higher Biology. Well done for finding it and getting here. We're going to have a wee look in this lesson at just diving right into the first part of the theory of Unit 1, Cells and Proteins. As I've said already, the first section of Unit 1 is actually laboratory skills for biologists, which you'll learn about when you're in Mr. Collier's class, as he's going to go through most of the practical techniques with you. As a result, we get to do all the fun stuff, so we're going to have a wee look today at finding out about what we mean by the proteome. So previously in Higher and National 5, you'll have covered some stuff on protein synthesis and gene expression. We're going to delve a little bit deeper into that and look at what we mean by the proteome, and then we're going to move on to having a look at membranes and membrane proteins. So your first task for you to be able to do is define what is meant by the word proteome, explain why the proteome is larger than the genome, explain why not all genes are expressed as proteins in any particular cell type, and be able to list the factors influencing the set of proteins in a cell type at a specific time. So you should remember that we have what we refer to as the central dogma of molecular biology. This is the fact that within every cell we have DNA, which is transcribed into RNA, which is then translated into forming a protein. However, although the genome of an organism is large, not all of the DNA is actually expressed as protein. Only around 4% makes it through this entire process. So what happens to the rest of it? We have some DNA which is not transcribed, and that can take the form of regulatory sequences that control gene expression. It can be the telomeric ends of chromosomes which are used for protection, or it may be junk DNA. Now, you don't need to know specifically what all of these sections do, you just need to know that untranscribed DNA might take any one of these forms. We also have, as I said, about 4% of DNA which is transcribed to form our total amount of RNA. However, not all of the RNA is actually then translated into coding RNA and produces a protein. We also have some non-coding RNA forms. So our non-coding RNA might take the form of tRNA, which you'll know from gene expression and protein synthesis, rRNA, which is part of the ribosome, and there may be either eukaryotic or bacterial forms, which again, similar to our regulatory sequences in DNA, will help control gene expression, and we'll find out a little bit more about these forms later on. Of the RNA which is translated, we wind up with something called the primary transcript, which you should remember from higher biology or higher human biology. That primary transcript is then converted into our mature transcript through alternative splicing. So we'll just have a quick recap on what we mean by alternative splicing. When we're looking at alternative splicing, what we're looking at is the difference between introns and exons and removing those non-coding parts. If this is our primary RNA transcript and we've got our coding sections numbered 1 to 6 along here, then depending on how we remove our non-coding regions and splice our coding regions together, we can wind up with several different proteins. So as you can see in this one, we've missed out our coding region number 3, over here we've missed out 2 and 4, and over here we've missed out number 2. Obviously within an actual coding gene, the RNA transcript would be much longer, so you've got many more options to be able to have to create different proteins. After alternative splicing, we've got our mature transcript. That is then created and turned into our polypeptide. So our transcript is then going to be taken to the ribosome, translated, and turned into a long chain of amino acids. From there, we can then add in what we refer to as post-translational modifications. And that's things that can happen to the protein after it's already been turned into a long chain of amino acids. So these post-translational modifications eventually will give us our protein. So as a result of both of these impacts, this alternative splicing and our post-translational modifications, it is possible to wind up with several different proteins from one small gene. Post-translational modifications take two different forms. First of all, there's the addition of other molecules, such as a phosphate or a carbohydrate, and then there's covalent cleavage. When we look at adding other molecules, you don't really need to know too many of these, but being able to remember one or two examples would be a good idea. So we have our nucleus where our DNA has been, our mRNA transcript has been produced, and our mature mRNA transcript has moved into the ribosome, where it's been converted into a protein. From there, that protein, our long chain of amino acids, may get additional molecules added to it. So we may have a phosphate added to it in the process called phosphorylation, a carbohydrate added to it, a ubiquitin molecule added to it, or a sumo molecule added to it. There are several other options as well, but as I say, you don't really need to know too many of the specifics of this, just that adding additional molecules is a form of post-translational modification, and that this might take the form of adding phosphate, carbohydrate, and probably worth remembering the ubiquitin molecule as well. The final post-translational modification that we may have is the proteolytic cleavage. So this is essentially when you've got your long chain of amino acids which have been produced. As a result of the different forms of amino acids, which we're going to learn quite a bit about in the next few weeks, then they have different molecules available for different levels of binding. What can happen? And this happens in the molecule insulin, for example. This could be pro-insulin here, and it needs to be cut in order to form insulin. So as a result of these binds, it's formed itself into a specific shape. And we've got these bonds formed between this section and this section here, which would be these ones over here and these ones down here. What can then happen is that the molecule could be cut here, and it could be cut here, resulting in a much smaller protein. And in the case of insulin, our pro-insulin molecule is cut to form insulin itself. If this molecule wasn't cut, it could remain as pro-insulin or as a slightly different form of the protein to do something else. So we have different alternatives to be able to make different types of protein. The proteome itself is referred to as the set of proteins in any cell at any given time, and can be made much, much larger than the genome, really as a result of all of these processes. We can have different DNA being coded into RNA, the total RNA then being coded for in different ways, the alternative splicing and addition of molecules and proteolytic cleavage as post-translational modifications will give us lots of different types of proteins. This means we can go from the genome to something that we refer to as the transcriptome, so that's our transcripts, primary and mature transcripts, and then we wind up with our proteome. So from a smaller genome, 22,000 genes, we can end up with over 1 million different proteins, and that's going to take the form of our proteome. So all of the different proteins produced by a cell at any given time. This is particularly important as not all cells are going to be coding the same DNA. So within a muscle cell and within a nerve cell, we have identical genomes expressing very different proteins. This allows cells to become specialised and supports the tissues and systems of which they're part. So our muscle cell is going to be expressing specific proteins relating to muscle tissue and relating to the purpose of that particular muscle cell. Our nerve cell will be expressing specific proteins relating to nerve tissue and relating to the particular purpose and function that it has. So although the genome from within each nucleus of each cell is identical, the proteins produced are going to be specific to that type of cell. It's also worth being aware that not only is genome expression differential, it is also responsive. So the set of proteins that are expressed by a cell type can change over time and are dependent on different conditions. So if we were to look at our nerve cell, as we said, it's got our identical genome, it's expressing its specific proteins. However, should there be any form of disease having an impact on the cell, that will affect the types of proteins that it can express. Should there be any signalling molecules around in its environment, that will change the proteins that it's going to be expressing at a given time. The metabolic activity and the cellular stress surrounding the cell, again, will have an impact on what types of proteins that cell is going to express. So you should be able to state that the proteome is the total of all the proteins expressed by a cell at any given time, that it's larger than the genome as a result of alternative splicing of our primary transcript into mature transcript, and also as a result of the post-translational modifications that we have in the cell. That not all genes are expressed as proteins in a particular cell type, because each cell type needs to produce specific proteins in order to support the function of the tissue that it's in, and that the factors influencing the set of proteins in a cell type at any specific time could be disease, signalling molecules, cellular stress, or metabolic activity.